So, hope that all of you are having a great day. So, we have started with the chapter cell. So, before we continue the chapter, just give me a thumbs up that voice is clearly audible to all of you. Just give me a thumbs up if my voice is clear and there is no glitch. Okay. So, yes, I can see all of you can hear me very clearly. So, fine. So, let's start. So, let's start with the class today. Now, here we have started with the chapter cell. So, we have started with the chapter cell, in which we have seen in the last class first about cell theory, which was given by which was given by Sladen and Schwann, and which was completed by Rudolf Virchow. Then we have seen the shape of the prokaryotes, which included four main type of shape: cocci, bacillus, spirilla and vibrio. Then we have also seen shape of different types of eukaryotic cell and their different sizes. Followed by, we have solved some question, and then we have compared prokaryotic and eukaryotic cell. Then we have talked about the cell envelope. We have talked about the cell envelope, which included the capsule, cell wall and plasma membrane. Then we have done some question on cell envelope, and then we were talking about mesosome. So, last topic we have discussed is the mesosome. So, good evening all of you. Now, no need to write anything in the chat box. Just continue with the lecture and we will learn, and whenever I will ask you question, just you need to answer. Till then, just focus on what concept we are reading. So, all these concepts are very new to all of you. You must be reading about mesosome for the first time. Now, here mesosome, in the last class, as I said, they have enzyme. They have enzyme. What kind of enzyme are present? So, they have respiratory enzyme. Now, what kind of? They have respiratory enzyme. So, the question is, what kind of respiration they do? So, they do aerobic respiration. So, they do aerobic respiration. They are responsible for aerobic respiration. So, can we say mesosome is similar to mitochondria? Similar to mitochondria in prokaryotes. See, usually mitochondria don't have this, prokaryote don't have mitochondria. But who perform the function of mitochondria? It is the mesosome which perform the function of mitochondria in the prokaryotic cell. Then next is the chromatophore. Again, very new topic, right? Now, again new topic. So, first thing, here it is chromatophore. So, just like mesosome, which was an infolding of the mesosome, which was infolding of the membrane, in the same way, as you can see, chromatophore is also the infolding of the membrane like this. Now, what is present in the chromatophore? So, it is the pigment which is present in chromatophore. Now, what is the function of these pigment? So, the function of these pigment is photosynthesis, right? So, they have the same function as the chloroplast in case of plant cell. So, mesosome is just like mitochondria and chromatophore is performing the function of chloroplast, and they are found in photosynthetic bacteria. So, these 2 points are very much clear. So, they both are membrane infolding. Mesosome is storing the enzyme and chromatophore is storing the pigment. Chromatophore is helping in the photosynthesis, where mesosome is helping in aerobic respiration. Further, this also we have discussed in the last lectures about the cell envelope. So, here is the overview of the cell envelope. Now the outermost layer is called glycocalyx, which can be capsule or slime layer. The cell wall is made up of peptidoglycan, and according to the cell wall we can divide bacteria into gram positive and gram negative. Similarly, the innermost layer is the plasma membrane. So these are the three, all these three together is called cell envelope. Now here one important concept and three terms are there. First term, and all of you note it down because I know there is a huge confusion here, and in the entire botany and in zoology these terms will be regularly used for the entire coming 2 years. So these terms are very important, so just note it down. The first term is called protoplast. Second term is protoplasm. The third term is cytoplasm. Now the question is, what is the difference between all of them? What is a protoplast? What is a protoplast? How is it different from protoplasm and cytoplasm? So first thing, when you take a plant cell, and remove cell wall, when you remove cell wall from the plant cell, it becomes protoplast. Then if you remove, now if you take protoplast and if you remove cell membrane from it, then it become protoplasm. And when you reduce, now let's say protoplasm, if you subtract, if you remove nucleus from it, then it becomes cytoplasm. So now can we say that cytoplasm plus nucleus form protoplasm? Protoplasm plus cell membrane form protoplast, and the cell wall plus protoplast will form the plant cell. So these are the difference. You have to remember, all of you note it down. These terms are very important. It will always be used in your upcoming topics. Then, so if needed, you take a screenshot from here. Take a screenshot if needed. Then we have already talked about the envelope. On the surface of envelope in bacteria, there can be three things: flagella, fimbriae and pili. Understand this. So these are the three hair-like outgrowth of prokaryotes. And this, in this case, we are talking about bacteria. Now if a bacteria is motile, bacteria can be two types, either it can be motile or it can be non-motile. If bacteria is motile, then it will have flagella. And if bacteria is non-motile, then no flagella. Then there is no flagella. So understood here this point? Now this hair-like outgrowth can be flagella, fimbriae and pili. Now if you see them all together, then you can see the difference very clearly. Which one is the longest? Longest is the flagella. Which one is the shortest? Shortest is the fimbriae. Right? Which one is most in number? Highest in number. So it is shortest but most in number. And flagella is longest but least in number. So remember these 3 points. Now if I simple say the function, so flagella has the function of movement, right? It has a function of movement. Similarly, pili has a function of conjugation. What is conjugation? Conjugation is between when two bacteria transfer DNA from one to another. It is called conjugation. So the function, the conjugation is done by pili. And lastly, fimbriae. So fimbriae will help in attachment. It will help in attachment. So fimbriae will help in attachment. Understood this point? So now repeat with me, what is the function of flagella? So the function of flagella is, its function is movement, locomotion. What is the function of pili? It helps in transferring of DNA from one bacteria to another bacteria, called conjugation. Third one, fimbriae. It helps in attachment to the substratum. Now these are the main function. Now if we study flagella in detail, then see, flagella has three parts. Flagella has three parts. The first part is called basal body, second part is called hook, and the third part is called filament. So we will call it basal body, hook and filament. Now if I ask you, out of 1, 2 and 3, out of basal body, hook and filament, which one is the longest? Tell me, which one is the longest among all the three? Basal body, hook and filament, which is the longest? So the longest one is the filament. Filament is the longest. Then if I ask you, which one is inside the membrane, or which one is inside the cell envelope? So basal body is present inside the cell envelope. Inside the cell envelope. If I ask you, which one connect flagella and basal body? So flagella and basal body is connected by hook. And it is shown here. See, this is the basal body, this is the hook, and this is the flagella. Flagella is the propeller, means actually this will move. But from where the energy will come? So basal body act as the motor, and this will rotate, and this will lead to the movement of bacteria. This point is clear. So now we have seen that the envelope of bacteria can have three types of hairy outgrowth. One can be flagella, another can be pili, another can be cilia. Now question here, is flagella present in eukaryote? Yes or no? Tell me, is flagella present in eukaryote? So the answer is, flagella is present in both eukaryote and prokaryote. It is present in both prokaryotes and eukaryotes. It is present in both prokaryote and eukaryote, flagella, but their structure is different. Flagella in prokaryote, it is made up of which protein? Flagellin protein. It is made up of flagellin protein. Whereas in eukaryote, it is made up of tubulin protein. We will see that in eukaryotes. So this is all about the flagella. Now this is the question. Now this is the question time. Most of you are connected here. Approximately 250 are connected. Now all of you answer it. This is a simple question which we have done. We have just seen it. Let me see who can answer it first. So you have to match it like this. You have to write your answer like 1 will go with, 2 will go with, 3 will go with, 4 and 5. All 5 you have to answer. Now here I am giving you the timer. I am giving you the timer. You have a time of 20 seconds here in this question. Your time starts now. Write all in one chat, you write all your answer. In one single chat, you have to write all your answer. Yes. So here, correct. So here first, flagella. So flagella will help in movement. Pili will help in DNA transfer. Correct. Fimbriae will help in attachment to the surface. Conjugation is the main function of pili. And which one is longer and fewer? So longer and fewer is flagella. So this is the correct match. So you can take a screenshot of it. It is very, very important. This one is very, very important. So keep it in mind. Now moving ahead. Now the ribosome is present in prokaryote as well as eukaryote. But before that, let's understand one more important point. Let's understand one important point, that is cell organelle. If we say cell organelles, so cell organelles are of mainly three types. Cell organelle are of mainly three types. The first one is without membrane. Cell organelle is without membrane or with single membrane. With single membrane. And lastly, with double membrane. With double membrane. So if we say without membrane, what are the cell organelle without membrane? So it is ribosome. Ribosome are the cell organelle without, and it is present in prokaryote and eukaryote. Then the second one is centriole. Now centriole is present in eukaryotic and especially eukaryotes and especially in animal cell, not in plant cell, right? These are, now further you can say inclusion bodies. We will see that inclusion bodies. So these are cell organelle without membrane. Now cell organelle with single membrane. So here you will see ER. Here you will see Golgi apparatus or Golgi bodies, right? Then you will see lysosome. Then you will see vacuole, right? Then you will see microbodies. So all of these are single membrane bound. Double membrane, three cell organelle, mitochondria, chloroplast and nuclear or nucleus, you can simply say. Nucleus also have two membrane, outer and inner membrane. So this again is very important. From here, how question will be asked? The question will be asked that how many of the following are single membrane, how many of the following are double membrane, how many of the following are without membrane, right? These kind of question will be asked from here. So you can take a screenshot of this also. It is very, very important, right? Question can be asked from here. Then, now moving ahead. Now coming to the ribosome. So we already know that these two category of cell organelle will be absent in, it will be absent in the prokaryote. So prokaryotes have only cell organelle without membrane, whereas eukaryotes can have cell organelle with single membrane or with double membrane. Now, so prokaryote, so ribosome is without membrane. Who discovered ribosome? So it was discovered by Palade. So that's why it is also called Pallidae particles. Now, these ribosomes in prokaryotes, they are 70S. Now we know that ribosome can be of two types, 70S ribosome and 80S ribosome. In prokaryote, the ribosome is 70S and it has two subunit. One is 50S, another is 30S. 50 and 30. So now the question is, this 50S and 30S, how they two are becoming 70S? Is there any error in the addition? Right? 50 plus 30 should be 80. How it is 70? Right? So for that you have to understand that these two subunits are not exactly attaching to each other. Actually, they are overlapping each other. So in between these two, 10S overlaps. That's why when you add both of them, you will only get 70S. So first thing, each ribosome has a larger subunit, and it has a smaller subunit. The smaller subunit in prokaryote is 30S and the larger subunit is 50S. This ribosome is made up of two parts. One is called rRNA, which is called R for ribosomal RNA. So it is called ribosomal RNA, and it is also made up of proteins. Right? So rRNA and protein make the ribosome. Ribosome have two subunit, larger subunit and smaller subunit. What is the main function of ribosome? So the main function of ribosome is protein synthesis. Right? It is the protein synthesis. They help in formation of protein. So that is protein synthesis. So main function of ribosome is protein synthesis. Now here you can see one structure. So this structure we will call it as polysomes. Right? This is polysomes. It is also called as polyribosomes. What do you mean by this? So here if I label it, see, this one is the mRNA. So on one mRNA, mRNA stands for messenger RNA. mRNA is used in formation of protein. So on a single mRNA, several ribosomes are attached. You can see multiple ribosomes are attached on a single mRNA. That is called polyribosome or polysomes. Polysomes are found only in prokaryotes. Polysomes are found in prokaryotes. So if question is asked, what are polysomes? What are polysomes? So polysomes are several ribosomes attached on a single mRNA. When is this polysomes formed? Polysomes are formed during protein synthesis. Later on we will see this protein synthesis is called translation. Protein synthesis is called translation. So overall understood every point of ribosome. I have covered each and every point in ribosome. First thing, ribosome is discovered by George Pallidae. That's why it is also called Pallidae particle. It has no membrane, so it is membraneless. It is membraneless cell organelle. It is found in both prokaryote and eukaryote. It is made up of rRNA and protein, larger subunit, smaller subunit. Several ribosomes on a single mRNA form polysome during the translation process. So this is all about ribosome. Now, question about ribosome. Very important question about ribosome. All of you see this. If I ask you about ribosome, ribosome in prokaryote are different from eukaryote because both prokaryote and eukaryote have ribosome. Ribosome is present in both prokaryote and eukaryote. Both have a ribosome. So, what is the difference? Yes, I am waiting for their first answer. What will be the answer? Ribosome in prokaryotes are. Ribosome in prokaryotes are. Some are telling A, C. Okay, A, C, A. Okay, Reshma is saying A, Yashwant, B, Afseen, C, A, C, A. Why C is coming? Why is many of you are answering C, made up of cellulose? What cellulose has to do with ribosome? What cellulose has to do with ribosome? Right? I hope you are not answering the previous question. Right? So, here there is no relationship of ribosome with cellulose. So, C will not be the answer. Right? So, ribosome in prokaryotes are different from those in eukaryotes. See, remember, prokaryotes have 70S ribosome and eukaryotes have 80S ribosome, right? So, one is 70S, another is 80S. So, tell me, prokaryote is smaller. So, prokaryote are smaller in size. So, A is the right answer, right? Praveen, Suresh, they have given A. Jashwant, A. Verma, A. Correct. Sriram, check your answer, Sriram. Harsha, fine. Mr. Farmer, you are telling D as the answer. What is the meaning of D? Lasya, A, fine, A. Very good. So now I think that you all understood A is the right answer. Fine. So now let's move ahead. This one, term polysome refer to. Next question, the term polysome refers to. Let me see how many of you can answer this. We have just seen it. Polysomes. Polysome is single ribosome, multiple ribosome. Priya is telling B, Sheikh B, Kalyani B. Okay, very good. Chetna B, Reshma B. Okay, so I am getting B as a lot of answer, but in between some of you are putting A also as the answer. See, the term polysomes. Poly means many. Poly means many, and zomes means here means ribosomes. Right? So that's why polysome is also sometimes called as polyribosome. So here it means multiple ribosome attached to a single mRNA strand. Right? So B will be the right answer. Right? B will be the right answer. Yes. RX, B is the right answer. Verma, B. Praveen, B. Dhanalakshmi. Right. Dhanalakshmi has written 20 times B. Very good. So B is the right answer here. Now, let's move ahead. So, one of the cell organelle without membrane we have seen is the ribosome. Another cell organelle we have seen is shown here, and that is called inclusion bodies. Inclusion bodies. Now, inclusion bodies are sometimes also called as storage bodies. They are also called as storage bodies. They are also called as storage bodies. So, see here, inclusion bodies, they have reserve material in prokaryotic cell. It is stored in the form of inclusion body. That's why it is also called as storage bodies. Now, these inclusion bodies are free in the cytoplasm and not bound to membrane, means these are membraneless. These are membraneless. These are membraneless. And what are the three types? The three types are, remember, very common, given in NCERT: phosphate granule, Cyanophycean granule, and glycogen granule. These three types of granules are important. Remember, phosphate granules, Cyanophycean granules, and glycogen. Previous year NEET question was there. Which of the following is an example of inclusion body? And these all were the options. Then another point, gas vacuole. Gas vacuole are found in blue-green algae and purple-green algae, photosynthetic bacteria. Now you will say, sir, just now some time back you said that vacuole is a membrane bound, and you said membrane bound is absent in prokaryote. So how can gas vacuole be present in prokaryote? So remember this point. Gas vacuole is not a true vacuole. Note this down. Clearly written in NCERT. Gas vacuole. It is not a true vacuole. True vacuoles. And that's why gas vacuole is also known as pseudo vacuole. It is also known as pseudo vacuole. So we say that it will also be membraneless. So if question is asked whether gas vacuole is with membrane, without membrane, so you will say it is gas vacuole is without membrane. Is it clear? Now where is gas vacuole formed? So gas vacuole is found in the photosynthetic bacteria. Photosynthetic bacteria. And these photosynthetic bacteria can be blue-green algae, purple sulfur bacteria, green sulfur bacteria, and these all are photosynthetic bacteria. So remember this point. Inclusion bodies, they are the storage body, and gas vacuole is an example of inclusion bodies. Gas vacuole are not a true vacuole, and three example inclusion bodies are phosphate, Cyanophycean, and glycogen granules. Then next, so this we have seen about the membraneless cell organelles. See, if you see prokaryotes, no, prokaryotes are very simple. They have this three cell envelope. Inside is the inclusion bodies. They have just ribosome. Then they have the cytoplasm, and then they have the genetic material. So now here if we see, all part is covered. Now the genetic material is left. So the genetic material, the genetic material in bacteria or prokaryote can be of two types, right? First is called the genomic DNA. It is called genomic DNA. As you can see here, this bacterial chromosome which is represented is called genomic DNA, right? This genomic DNA is present in a structure called nucleoid. Genomic DNA is present in a structure called nucleoid. It is present in a structure called nucleoid, and another DNA is called the plasmid. Another DNA is called the plasmid. Now, so one is genomic DNA, another is called plasmid. Another is called plasmid. Now, both of them are double-stranded DNA. Both of them are circular DNA, and both of them are naked DNA. Means naked DNA means they are without histone. They are without histone. No histone is present, that's why they are called so. Is it clear? So if question is asked the genomic DNA, so you will say they are circular. This genomic DNA, it is circular. Is it double stranded? Yes, it is double stranded DNA. And is it without histone? So yes, it is without histone, right? Also one more thing, if somebody asks you how many chromosome is present in bacteria, so your answer should be each bacteria has a single chromosome. Each bacteria has single chromosome. Whether it is E. coli, Bacillus, Lactobacillus or any bacteria, every time number of chromosome will be single. Again, this is given in NCERT that genomic DNA is double stranded, circular, and it form a single chromosome. Is it clear? Now this plasmid is the extranuclear DNA, and plasmid has some specific gene which is responsible for antibiotic resistance. So remember this point also, plasmid have some antibiotic resistant gene. Is it clear? Now, so this is about the genomic material in the prokaryotic cell. Now if we see, this is the overview of a cell. This is a typical bacterial cell. Let's once see all the part of it which we have already discussed. So the first point, the outermost envelope. So slime layer represent the glycocalyx. Now it can also be capsule, then cell wall and plasma membrane. So all these three together represent the cell envelope. Inside the cell you see ribosome is present. Then in the membrane you have respiratory enzyme present in the, as the mesosome. Then on the membrane you have flagella, then you have fimbriae, then some pili can also be present, right? Here along with mesosome, chromatophore can also be present, chromatophore. Then inside will be inclusion bodies. Inclusion bodies can also be present in the form of gas vacuole. Apart from that, the main DNA, which is the nucleoid, which will have the genomic DNA. Now these ribosome can also form polyribosome, or just we have seen polysomes, right? Polysomes. Apart from DNA, they have RNA also. Then they can also have some circular DNA in the form of plasmid. They can also have some circular DNA. So this is overall all the cell part of a prokaryotic cell. You can take a screenshot in a single diagram. All points are mentioned here. Now in the beginning only, when we talked about kingdom Monera, we have talked about Mycoplasma. So all of you recall and tell me what is special about Mycoplasma. I am waiting. I will read all the response, whatever you will write related to Mycoplasma. So write related to Mycoplasma only. Whatever you can recall. What is the special thing we have talked about Mycoplasma? What is the main function or main character of Mycoplasma? I am waiting. Let me see who answer first. About Mycoplasma, we have studied it in kingdom Monera. Yes, Tanish has said it is smallest bacteria. Very good. Lasya says smallest cell. Suresh, smallest cell. It can survive without oxygen. Pooja, very good, very good. Smallest cell, smallest cell, smallest cell. Okay. Apart from smallest cell, apart from smallest cell. Okay, smallest cell. All of you are writing smallest cells. Think a little more about it. Right? Think about that. So I will give you one hint. Like to think about it. Think about cell wall, think about cell membrane, think about the outer covering. Can survive without oxygen, Kalyani has said. Okay, smallest cell, again smallest cell is coming, fine. Okay, size, okay, very good. Sanmukh has given the size, 0.1 micron. Sanmukh, 0.1 micron is the size of PPLO, which is a specific type of mycoplasma. In general, if you have to write the size of mycoplasma, you will use the 0.3 micrometer, right? In general, mycoplasma is 0.3 micrometer. Okay. Anita said it is anaerobic. Lacks cell wall. Khalil has said it lacks cell wall. Kalyani also said it lacks cell wall. Very good. Very good. So now you are, okay, Vijay is telling it is double member. Is it double membrane? No, it has only one membrane. Lacks cell wall. Yes. Yes. Chalam D is telling jokers. Very good. So tell the complete. Okay. Yamala is telling completely. Jokers of microbiology. Very good. So now I think all point is yes. Misbah is also telling, also called jokers of microbiology. Very good. Yes. So all points are now covered basically. So mycoplasma we talked about, it is the smallest cell. Right? It is the smallest cell and its size is 0.03. But one very important part of mycoplasma was it lacks cell wall. Now because it lacks cell wall, it is called jokers of microbiology. It is also called jokers of microbiology. Clear? Now these mycoplasma can survive without oxygen. They can also survive without oxygen. Without oxygen. So can we say that mycoplasma is the only prokaryote without cell wall? If question is asked, name the prokaryote without cell wall. So your answer will be mycoplasma. Now moving ahead. So this is about mycoplasma and overview of the prokaryotic cell. Now we will see some question. Now these questions are in general related to prokaryotic cell, which we have studied till now in all the class. Till now in this entire chapter. So first question. All of you start answering it fast as possible. These are very simple question. Okay, Lashya is adding one more point about mycoplasma, which is very interesting and very good. Lashya, sir, it is resistant to antibiotic. Very good. But what kind of antibiotic? The antibiotic which target, which target cell wall. So it is correct point. Which target cell wall. Yes, I am getting the answer. Prokaryotic cell lack nucleus. They lack nucleus. Instead of nucleus, what they have? So instead of nucleus, they have nucleoid. They have nucleoid. Then prokaryotes are typically characterized by, tell me. Second, what is the answer for second? Prokaryotes are typically characterized by well-defined nucleus. No, well-defined nucleus is not present. Presence of membrane-bound cell organelle. No, it is also absent. Single circular chromosome. Single circular chromosome is also called plasmid. Right? So plasmid is present. So correct answer will be C. Correct answer will be C. Very good. Pooja, Praveen, Sheikh. Very good. Now next Next question, third question. Which of the following is absent in prokaryotic cell? Which of the following is absent in prokaryotic cell? Again, very simple. Ribosome, yes, it is present. It is 70S. Plasma membrane present. Cell wall present. Nucleus not present. So, nucleus will be the right answer. Third, B will be the right answer. Yes. So, Sheikh, Chandra, Lashya, very good. So, you are giving right answer. Next is prokaryotic cell reproduce by. So, option D is hidden. D option is budding. Budding. So, do they reproduce by mitosis, meiosis or binary fission? Remember, recall kingdom Monera. In kingdom Monera, we have discussed about it. Prokaryotic cell, that is bacteria. Bacteria divide by which process? They divide by binary fission. They divide by binary fission. Then fifth question, the DNA of prokaryote is found in the, where is DNA found? DNA in prokaryote is found in which region? Very important, interesting question. Where is it found? Is it found in nucleus? No, nucleus itself is not present. Is it found in mitochondria? Not present in prokaryote. Golgi apparatus not present in prokaryote. What is present? Nucleoid region is present. 5, correct answer is B. Very good. Sheikh, B. Jaswant, B. Tanish, B. Very good. So B is the right answer. B is the very good right answer. Now next one. So as I have promised that every day we will do 2 assertion reason questions. So the today's assertion reason question. First, so all of you, show me that you all are very comfortable in assertion reason question. Tell me what will be the answer. Prokaryotic cell do not have membrane bound organelle. The cell structure in prokaryote are not compartmentalized. What should be the answer? See, prokaryote cell do not have membrane bound cell organelle. Is it correct or not? Yes, assertion is correct. The cell structure in prokaryote are not compartmentalized. If membrane is not present, compartment is also not present. So this is also correct. Who is compartmentalized? Eukaryotes are compartmentalized. So both assertion and reason is correct, and reason is the correct explanation of assertion. Is it clear? So first, A will be the right answer here. Next question, tell me. Ribosome in prokaryotes are smaller than those in eukaryotes. Prokaryotes, ribosome is smaller than eukaryote. True or false? So it is correct. Ribosome of prokaryote is 70S. Eukaryote it is 80S. So yes, this is smaller. Next, prokaryote ribosome are of 80S type. Prokaryotes are 80S. No, it is 70S. Right? So what is correct here? Assertion is true, reason is false. So correct answer for this will be C. C will be the right answer here. Yes. Sagar, correct. Praveen, correct. Very good. Reshma, correct. Faiz, correct. So all of you, very good. So well done. So now you all are getting very much comfortable with assertion reason question also. Assertion reason question also you are all are getting comfortable. So now we will stop here. With this, the prokaryotic cell is done. In the next class, we will start with the interesting types of or interesting characteristic of eukaryotic cell. So till then, keep reading NCERT, and all of you just revise whatever we have done till now in prokaryotes. So thank you all of you, and a very good evening.